Oh, what's up? Alright, I should never do that again, right? Cool. Alright, hey, what's up? Me, and you know who I am. If you've been watching my videos for the last few days, you know that I'm the guy who's doing daily videos on the search for eternal life. So I don't think I explained well what my impetus and motivation was for this before. Okay, so anybody who knows David Sinclair, who knows who he is? I mean, if you're watching this video, chances are that you've run across him at least once, okay, in the past. At least his name, you've seen videos of him. It's this guy here, okay? Tons of Google images, okay? That's him from 2017. He's like 50 years old also, or 51 or 52 years old, okay? Looking pretty good for his age, okay? That probably is in line with one of his whole thing, which is slowing the aging process, okay? So if you look up his Wikipedia articles, he's talked a lot about sirtuins, NAD, and the epigenetics of aging, okay? So what are all these words? They're different words, and we'll get to them in our course of biology, but epigenetics is really one of the more interesting ones, okay? So it has to do with, if you know what epigenetics is, then feel free to mute this video. But I'm interested by the idea of epigenetics. Okay, I'm not going to sign up. And epigenetics is basically what happens when. Okay, all right. So this is a little embarrassing, but I hit a little oopsie. Okay, while filming this video. Anyway, I was talking about epigenetics. Okay. So what is epigenetics? Epigenetics is basically things that happen to you biologically that are not a result of mutations in your DNA, but rather changing of your epigenetic genome. Okay, so epigenetics is something that doesn't, it's a process that won't affect your germ cells, but will still affect your children, because your non-germline DNA was affected, and thus you can pass these things along to your germline. Now it's a pretty complicated subject, and I'm going to get a little deeper into epigenetics later on, but let's just say if you want a quick chemical taste of what that is, you can look into DNA methylation. Okay, so anyway, let's not get really into epigenetics right now, but let's talk about, I was talking about David Sinclair, okay? So why was I talking about David Sinclair? Okay, well, I had a reason, because I wanted to give you a little bit of background into why I'm doing these videos, okay? DuckDuckGo, great app. Because my idea is that I want to make the search for eternal life something that's public, okay? I'm not a biologist by trade, okay? So you probably know that, and somewhere down the line, I'm going to be confident enough to say that I'm an expert biologist, because that's where I'm trying to get in my life, and particularly specifically into bioinformatics and into the search for eternal life. But I have a long ways to go. I'm in a bit of a career either transition's not the right word, augmentation, because I'm going to use my current technical skills to augment my biological. I'm going to use my current technical skills, and I'm going to augment those with these that I'm learning from my biological studies here to help me search for the meaning, the Ponce de Leon, the fountain of life. Okay, Ponce de Leon was the guy who was searching for the fountain of life, I believe, and he died in Florida in the 1500s or something. I don't know, I don't remember exactly, but either way, yeah. So let's get moving, because I want to talk about the topic of today. Okay, so the topic of today is basically cell walls. Okay, what are cell walls? Let's come up with a picture. Cell wall. So a cell wall, from your high school biology, I didn't say sail wall, I said cell wall. From your high school biology, you should know what a cell wall is. Okay, a cell wall is the part of a plant cell that, and it's a part of several animal cells too. I mean, cell walls are actually found throughout the kingdom of life, but it's most commonly associated with the plant cell, and it's the part of the plant cell which provides structural support to the plant. Okay, it prevents, it allows for rigidity of the plant, and it allows for, it prevents, consumption of what's in the insides very easily. It gives it its thickness. I've talked about turgor pressure before. It allows the plant to essentially withstand pressure of water flowing through it and dripping out through the stoma that allows the plant to stand upright. It's basically the component of the quote skeletal system of a plant. It's also very important for many other things, including the transfer of substances throughout the plant. Plants are not mobile, so they need the cell wall to give it, structure. Okay, like we have our skeletons to give us structure, which are designed for the purposes of structure while moving. So the cell walls are designed for structure while stationary for plant cells. Okay, bacteria also have cell walls as well. You can look at bacterial cell wall, and bacterial cell walls, I mean, they have a similar structure to bacteria. Okay, you can see bacterial cell wall, bacterial cell structure. We can probably find the cell wall, but see, the cell wall and the cell capsule, okay, are two external structures to the bacteria. Okay, so this is interesting. I'm taking a little diversion here. I'm talking about cell walls in general, but cell walls are basically composed of polysaccharides mostly. Okay, and different kinds of polysaccharides. Cellulose is probably the more commonly heard one that you've heard of, and cellulose is structurally a series of tubes, right, like the internet. Okay, I keep using that joke over and over again, but for the picture to help make sense, cellulose looks like this, and man, my internet is blazingly fast tonight. I don't know why. It's probably because of where I'm located right now. But okay, let's take a look at cellulose. Okay, this is a cellulose microfibril. Okay, this means the cellulose microfibril is the basic unit of a cellulose structural component, right? So all of these are mashed together within the plant cell wall. Okay, and these microfibrils, if you take a look at the microfibril structure of a cellulose, if you were to zoom in, you'd see many strands of cellulose bonded together parallelly into this microfibril, like hundreds. And there's microfibrils, macrofibrils, etc. But these are all bound together. And they are, cellulose is very strong. I talked about how, at some point, the reason why the strength in cellulose is present, because essentially cellulose is straight chain glucose, right? So if you look at if we go back to cellulose, this is the structure of cellulose, right? So you have these straight chain, these are all glucose molecules, alternating alpha beta glucose molecules. Okay, I could be wrong about that actually. I know they're glucose molecules, but I believe there could be 4,6 versus 6,4 versus 4,6 alternating. Okay, so basically these glucose molecules, right? Like you can tell where the glucose structure lies. Okay, and these straight chains are cross-linked to each other via these hydrogen bonds. Okay, these hydrogen bonds form because of the alcohol group, the hydroxyl group interactions between these glucose molecules, and the hydrogen bonds give the cellulose strength, right? This lateral strength here, and it can resist shear. because of these, okay? So, and these are polar, right? Everyone knows how hydrogen bonds work. So you should understand that these are polar bonds, and they're essentially electromagnetically bonded together, okay? So hydrogen bonds, that's what makes cellulose really strong, okay? Now, these are basically straight chain polysaccharides, okay? This is glucose, 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 glucose in a chain. This can be contrasted with the structure of glycogen, which actually has a structure like this, glycogen, okay? Glycogen has a matrix structure, matrix polysaccharide structure. You've probably heard of glycogen before, but glycogen is a polysaccharide, and it's one of the primary structural storage mechanisms of energy within a cell. It even says it in this little Wikipedia thing here, but it's taking too long to get to, so I guess we're not going to get to it tonight. So let's continue on with cellulose, okay? So interesting thing about cellulose. See, this is hydrogen bonds within the strands. Natural cellulose is cellulose with structures I alpha and I beta. Cellulose produced by bacteria and algae is enriched in I alpha, while cellulose in higher plants consists mainly of I beta. So there's different kinds of cellulose. Now, going back to bacterial cells contain a cell wall that's not necessarily cellulose. It's not all sugar. Okay, and when I say it's not cellulose, there are polysaccharide components, but there's other components of the bacterial cell wall. For example, there's peptidoglycans and other types of layers, of semi-permeable layers, which give bacteria strength, and ability to withstand osmotic pressure gradients within the cell. Because without the cell wall, bacteria, remember, are on their own, and they need to have a very strong kind of structure to withstand being crushed, right, or exploding. Okay, so the cell wall prevents that. Actually, it's more like, the way that plants take in water, right? The way that plants use their cell walls to get structure is what I mentioned before, was called the application of the Turgor pressure, okay? Here's glycogen, okay? Finally it came up. So glycogen has an organization that's similar to kind of it's a branched polysaccharide, okay? Branched because glycogen, these are glucose molecules, and they're not in cellulose. These are all chains that are parallel to each other. In glycogen, you can have a glucose molecule attached by an O-glycosidic bond to another part of the glucose molecule, another glucose molecule besides its 4 or 6 repeat monomeric bond attachment. So you don't create necessarily straight chains. And this matrix chain here, okay, this is what hemicellulose. This is more like the structure of hemicellulose, okay? This is the atomic structure of single branch of glucose units in a glycogen molecule, okay? So this is basically this macromolecule of glycogen kind of looks like this. So hemicellulose even has less structure. And it's not all consistent of glucose. Hemicellulose, now if we go back to our original diagram of cellulose, where is it? These are my older tabs from yesterday. I'm not going to talk about those. Hemicellulose, okay, looks like this. Okay, so you have many other things. These are all these are different sugars bonded together, right? You have mannose, glucose, galactose, xylose, okay? Mannose basically being this. You have these are all isomers of stereoisomers of glucose, meaning they have a similar structure to glucose. You can take a look. These sugars like xylose, mannose, glucose, that's this guy, and then galactose, and they look like all kinds of dancing sugar molecules, right? This is like the rockets of sugar, okay? This mannose has both of his legs up in the air, like a man. This glucose is like the leader of the group because that's the most well-known and prominent one. This xylose is kind of like a sidekick, kicking up with the left hand in the air. And the galactose is like a cheerleader praising the galaxy. But these are all the same structure, right? Mostly, like you can tell these are hydrogen bonded together and connected via these O-glycosidic bonds. These are what comprise hemicellulose. Okay, and hemicellulose, if we go back to the cell wall, hemicellulose is this red stuff here. Okay, this red hemicellulose stuff binds these cellulose microfibrils together, right? Because essentially this is called secondary structural support of the plant cell wall, and these microfibrils would kind of not necessarily be. I mean, one could think that they would be hydrogen bonding between each other as well, but I guess the pack structure of a cellulose microfibril doesn't allow for that, or I don't know all the details, but I guess hemicellulose helps with that, but it's weaker than cellulose. Hemicellulose can be hydrolyzed easily, and it says that somewhere here, that this is xylan. This is a type of hemicellulose, okay? And hemicellulose is in wood, okay? Beta-D-glucopyranose with carbon positions. Oh, I'm just reading random stuff. So, see, let's go to the natural functions. To me, when I see something like natural functions, that's what really helps me to understand something in these wiki articles. So always look for something about natural functions. Okay, you have hemicellulose here. This looks like a diagram a kid made. Okay, maybe it is. But this is kind of like, if you look at a cross section or a lateral section, right, you'll see cellulose, hemicellulose, and then you have this raminogalactic, whatever that is. That's also, I assume, necessary. Pectin is another very important structural protein that kind of binds this whole structure to the middle lamella, right, which is basically the space between the cells, okay, after the plant cell wall. So middle lamella, right, of the plant cell. Unlike cellulose, which is primarily found in the secondary layers, okay, the secondary layers, the middle lamella is like the primary layer. We can actually look that up and see what the lamella refers to as thin layer membrane of plate tissue. It's a very broad definition, can refer to many different structures, okay? And you can think of lamellae, right, as laminar, right? So it's like a plate, like laminar flow and fluid, okay? Then that's kind of what it means. A thin layer of organic tissue can be called a lamella, and there's a wide array of functions an individual layer can serve. So in a plant cell, in plant cells you have the lamella of the plant cell, which is kind of the top layer, and then it can be broken down into the middle lamella and the upper lamella and stuff like that. Middle lamella is a layer which cements the cell walls of two adjoining plant cells together. It's the first formed layer which is deposited at the time of cytokinesis. Okay, so that's important because when plant cells divide, what they do, they don't necessarily, they have a more complex cell division than eukaryotic cells, and they have a different type of cell division, okay? Because remember, plant cells are separated by cell walls, okay? So what do they have to do in that case, right? Like this, like you don't divide the cell during, what are the different stages of cell division. When those, I should know this like the back of my head, and I'm going to relearn this at some point, but after mitosis occurs, what happens is the plant cell separates into pinches off, right? But the thing is, a normal cell pinches off during mitosis into two different pieces. I'm making this pattern here, but a plant cell actually will not just pinch off and form the plant cell around it. This matrix of plant cells has to be, the plant cells are very communicative, and they rely on their neighbors a lot. And you can tell because of these plasmodesmatae right here. Pointing to it right now, or these what are also are known as intercellular channels, which allow for transmission of cytosolic cell between plant cells and other little molecules and whatnot, there is a required structure to the plant cell. So the cell, there has to be a planning going on. So what happens is after mitosis occurs, what happens is the plant cell wall gets deposited within the other plant cell, right? So the cell divides. Like we can actually, maybe we'll find plant cell division here. Let's hopefully find an animation. Okay, you can find a description of what the difference is between plant cell and animal cell division, but main difference between plant and animal cell division. Plant and animal cell division occurs as a part of a life cycle. Cell division in both plant and animal cells can be divided into two types: vegetative cell division and reproductive cell division. The vegetative cell division, which produces genetically identically two daughter cells, is called mitosis. Okay, that's standard. All things go through vegetative cell division. But then you have reproductive cell division, which produces 4 gametes containing half of the chromosomes. That's meiosis. Okay, so we are talking about vegetative cell division. Okay, so let's all focus on that. The key difference between plant and animal cell division is that plant cells form the cell plate in between the two daughter cells in mitosis, whereas the cell membrane forms the cleavage furrow in between the two daughter cells in animal cells. Okay, basically what that means is that when the daughter cells split from the parent cell in animal cell division, in vegetative animal cell division, when they split, the phospholipid bilayer kind of naturally forms in between those two, and they go their separate ways. But in plant, or they get stitched together, depending on how they form in the tissue. But in plant cells, the cell wall kind of structurally is like contained within the cell of the parent, right? And differentiation between the daughter cells is a little more complicated. But there's we definitely don't want to go into meiosis right now. Okay. But let me see if I plan my plant cell division video. Plant cell division. Okay. Images. Okay. Cell wall. Vesicles fill within cell wall component. See, this is where the cell wall is like vesicles fuse, forming new cell wall. These vesicles are emitted, form the new cell wall. The new cell wall being complete is like when a room is divided up into two things, and then you have the two plant cells. And look, notice the simultaneity of these processes, these mitotical processes. Telophase, right? You see, by the time cytokinesis fully occurs, where the cytosol is differentiated between the different animal cells and the phospholipid bilayer and the new plant cell is present, that's when the re-randomization of the chromatin occurs in the DNA, in this area, okay? Because you see that the chromosomes are straight length in the plant cell, and now they're all like random chromatin fibrils. So they don't have, they don't have the similar structure as they did during cell division, okay? And also there are less pores in the nucleus at that point when the cell wall is complete, when the plant has plant cell has divided, okay? So anyway, that's just something to think about, okay? Now, we've gone a lot into the plant cell walls. It's interesting to note that certain animals also have cell walls, right? Tunicans. I believe that's how you pronounce it, or say it, or spell it even. Tunicans. No. Not tunicans, the cat. Tunican. I'm looking for the animal. Animal. I thought it was pronounced like with a tunic. Oh no, I'm not looking for tunics. Okay, there's a tunican. Maybe it's a tunican. Tunican. Like tunican. Oh. Tunica. Nope, that's not what nope, that has nothing to do with what I'm trying to find. It's a sea animal. A sea animal. Oh, God. You know what? I could spend the entire time. The whole point is that some animals have cell walls, and tunicans are a specific type of animal that has cell walls. It's an undersea animal that looks like a sponge. And look it up if you have a chance. Anyway, okay, so I think that's about enough. I could talk a little bit more about hemicellulose or pectin. Pectin is another very important protein. Apparently, it's used in food. Not protein. It's an important. Oh, I think pectin is a protein. Let's. I should know this already. Pectin. It's a structural acidic heteropolysaccharide, meaning it's a sugar polysaccharide, but it's a heteropolysaccharide containing. So it's not all glucose connected together. Just like hemicellulose is not a single polysaccharide. It's several different types of monosaccharides connected together. So it's a heteropolysaccharide contained in the primary cell walls of terrestrial plants. Its main component is galacturonic acid, a sugar derived from galactose. It was first described in 1825 by Henry Branicott, way before I was born. Okay. You can see there's pectin. Pectin is also like one of those things that like they put in, it says right here, it's a gelling agent. So if you have like things like jelly or things like that, or gelatin, you have pectin in it. So I wonder, like when you have those gel caps in the store, how much are those gel caps are pectin? Those gelatin gel caps are pectin, and how much of those are pork? I don't know. I figured it was easier to come by pectin than to come by pork. But I don't know the gelatin capsule industry myself. Maybe I'll learn that one day. All right, I think that's enough for today. Hasta luego.